So proteins, right? So why can't we survive on just carbs and lipids? It's got enough calories in it, right? We would have enough energy, but there's something inside of proteins that we don't get in other places. So if you think about like carbohydrates, we have lots of carbons, we have lots of hydrogens, lipids too. We have lots of carbons, we have lots of hydrogens, we have oxygens. But one of the things that we really, really need that we don't get from basically anywhere else is nitrogen and sulfur. So nitrogen and sulfur we get in proteins. They're not really in carbohydrates and lipids. So if you look inside the cell, like we did in the video, most of the stuff that we saw was proteins. Like all the active was protein, all the myosin, all the microtubules, all of those things were proteins. The second most common is all the lipids, like the lipid bilayer, the layers on all the vesicles, all that kind of stuff. So having enough protein is really, really important. And what we're going to see is that proteins vary so much, right? And you kind of know this because you did 109. So you know how many different proteins there are. And the good news is I don't make you memorize the amino acids. So you get exactly the same sheet that you got in 109. You get all the amino acids. So we used to think, we're wrong, but we used to think that proteins were the genetic material of organisms because of the diversity, right? If you look at the most common amino acids that are out there, there's 20, right? There's more than 20, but the most common, there are 20. And we thought, well, the more diversity there is, the more information we can store. But it's not true. So even though DNA only has 4 different bases, you can store a ton of information in those combinations of those 4 bases. So, but what we understand now is that the diversity of proteins is super important because we need a big diversity of function. DNA, there's only one function: store genetic material, right? But proteins have lots and lots of different ones. So we know proteins are polymers of amino acids. Amino acid is the monomer, and then proteins is the polymer. So there are tons and tons and tons of different biological functions. So when we talk about enzymes, almost all enzymes are protein. There are some enzymes that are RNA, but we won't really talk about those. But you got to think about, there are some reactions that we try to do in the lab that take days, right? A really long time. Or you have to heat them to this insanely hot temperature to make them go. But can we do that in the body? No, we can't wait for days for a reaction to happen. Like our reactions have to happen instantaneously, like less than a second. And so the way that we make those reactions go fast is to use enzymes. Defense proteins, these are super, super important. Sometimes you'll see them called antibodies. Sometimes you'll see them called immunoglobulins. But it's the same thing. Antibodies is the same thing as immunoglobulins. And antibodies are always going to recognize antigens. So just like we talked about the antigens that sit on the outside of your red blood cells, makes you type A, type O, type AB, right? So those are the antigens. But we know that things like bacteria, viruses, those kinds of things have antigens. And so if we express antibodies, we can combat those proteins, that those bacteria, those viruses, those kind of things. We have transport proteins. So we have to make sure that we maintain a separation between the external environment and the internal environment of our cells. So we have to have transport proteins that will allow us to import and export things across that membrane. That's why we call it a semi-permeable membrane, because there are some things that will be transported in and out. Regulatory proteins, oh my gosh. Regulatory proteins are such a massively big category. They control everything, but for y'all, really, metabolism is big, reproduction is big, but they regulate basically. The structural proteins, these are my favorite of all time. Love them so much. But they keep the shape of the cell. So have you ever seen a neutrophil chasing a bacteria? You need to see that. Neutrophil. Yes, but like they actually move and chase. So this is the big thing, is the neutrophil. The circles are the red blood cells, and then the bacteria are diplococci. And so you actually see this neutrophil has to infiltrate tissues in order to get to the bacteria. And so eventually he's going to catch it. There he goes, caught it. Isn't that so cool? So there's not an external skeleton that's allowing that to happen. It's an internal skeleton. And the internal skeleton basically pushes. Internal. It's this internal cytoskeletal proteins that allow that to happen, which is why I think they're so cool. All right. So, structural proteins, movement proteins are like remember when we saw the video and it was the little motor protein that was swinging his legs around and pulling the vesicle? That's an example of a movement protein. Like in muscle, it's the myosin that pull on the actin in order to shrink the sarcomere. Did y'all do that in anatomy? Okay. So transport means in and out of the cell, in and out of the cell. So if you have a lipid bilayer, so this is our lipid bilayer, right? You have these proteins that are basically like little tubes that allow, let's say, glucose to go into the cell, right? Or sometimes it's like ions, like sodium, or maybe potassium needs to get out, or whatever it is, right? That's a transport protein. A movement protein is an example like myosin. It's inside the cell, inside the cell. Yeah, that kind of does this. Like, wait a minute. Yeah. And then the last category is nutrient proteins. And nutrient proteins, you got to think about like embryonic development. So I like eggs, right? But they're little baby chickens. Right? That was weird. I like eggs, right? Okay, so in an egg, right, the albumin in the egg, albumin is an example of a nutrient protein that's there for the developing embryo, but we eat it. In milk, it's the casein. Milk. Oops. The casein, casein, casein. Right, that gives protein to the developing, whatever it is, mammal. All right, so amino acids. So when we talk about, do we say? No. Okay. So when we say the name amino acid, right, it's not just that it's mean. The amino refers to an amine group. So what's an amine? With some number of either hydrogens or R groups. So, right? And sometimes if it's protonated, it's positive. Sometimes it's non-protonated, so it only has 3 bonds. but an amine group. And an acid, but what kind of an acid? Carboxylic acid. So we have two groups, the amine and the carboxylic acid, and so we call them amino acids. And so we call them, we're going to talk about why we call them alpha amino acids in just a minute when we look at the structure, but we got to look at the structure of that. So you can actually find way more than 20 of the known amino acids, both in nature and we can make them in the lab. So we say that there are 20, but that's a lie. There are way more than 20. But we kind of focus on the ones that are in very, very, very high prevalence, right? So when we call them alpha amino acids, the alpha tells us the location of the amine. So I'm going to show you. I'll show you on the structure. And we say 19 out of the 20 are stereoisomers. In order to be a stereoisomer, you need at least one chiral center, which means that all the amino acids have at least one chiral center except for one, and that is glycine. Glycine is achiral. And we'll see why. So if you look at an amino acid, here's an amino acid, right? The backbone of an amino acid, like starship. So the designation of the Starship Enterprise is the NCC 1701 ABCD, all those kind of things. So the backbone, this is how I remember it. The backbone atoms are nitrogen, carbon, carbon. That's where the NCC comes from. So there are two carbons. So we have to have a way to like talk about the carbons and say which one is which. So we have a carbonyl or a carboxylate carbon. That's the blue carbon, right? That's this guy. And then we have an alpha carbon. And the alpha carbon is the carbon that has the R group attached to it. So the alpha carbon is the chiral carbon, because the carboxylate carbon can't be chiral because it has a double bond, right? So the alpha carbon has to be the one that has the chiral center. And the one with the chiral center is the one that has the R group. And so you have the handout, you grabbed one, that has all of the amino acids posted. Yeah. So what's in the light gray shade, that's the backbone. So you see the NCC, and then what's in the darker shaded on top of it is the R group. So you see how the light shaded is the exact same for all 20 amino acids, which I should probably pull that up on here. Let's see, that came from my 109 class. Give me 1 second and I'll pull it up. I know I have it in my 109 class. Here we go. Here we go. So what's light here, this is the backbone. And then what's highlighted here is the R group. And so what you see for every single amino acid is that this portion here is different. The backbone is the same for all the amino acids, but what's different is the R group. R. Yeah. Okay. So we call them alpha amino acids because the amine group is bonded to the alpha carbon, not to the carboxylate carbon. So it's an alpha amino group and it's an alpha carboxylate group because they're both bonded to the alpha carbon. So that's why I call it an alpha amino acid. Yeah, it doesn't make sense until you look at the structure. So then look on the So we call this the N-terminus. N-terminus. So it's just the beginning. So the beginning of the amino acid is always going to be the N-terminus, and the end of the amino acid is always going to be the C-terminus. So this is the end. So all amino acids and all proteins have a beginning and an end. The beginning is always the N-terminus. The end is always the C-terminus. Okay, so we said the alpha carbon has a carboxylate group. We talked about that. It has an amino group, and the amino group is protonated. So it has 1, 2, 3, 4 bonds to it. So therefore, it's going to carry a positive charge. So it's a protonated primary amine. And if you're at physiological pH, which is close to 7, right? Very close to 7, the amino acid is going to look the way it does on the screen. It's going to look like a carboxylate and not a carboxylic acid, and it's going to look like a protonated amine, not a deprotonated amine. So if I change the pH, I can change the protonation state of the amine and of the carboxylic acid group, but we'll talk about that later. So because when we look at this molecule, let me erase some of this. It's getting kind of crowded. So if we look at this molecule, right, on the N-terminus, I have a positive charge, and on the C-terminus, I have a negative charge. So it's a neutral molecule at this pH, and we call that a Zitter ion. It's a ridiculous word. It's a ridiculous word, but would have been a better name than X, right? But anyway. Okay, so we know that we have those charge groups on either side. So if we change the pH, basically we can change the protonation state of those two groups. So let me show you. I want to show you. So our backbone is a nitrogen, a carbon, and a carbon. And the nitrogen is going to have either 2 or 3 things attached to it, right? But we said if we're at a neutral pH, then we're going to carry a positive charge on the nitrogen. We're going to have a hydrogen coming off of our alpha carbon, and then some kind of an R group. Our carboxylic acid group, we have to add our oxygens, and we're going to leave that oxygen without a hydrogen because we're going to say we're at pH 7, right? But okay, what if I don't want to be at pH 7? What if I want to get really, really acidic, right? I want to go down to pH 2. What's going to happen? If I'm in an acidic environment, do I have a lot of H plus or a lot of OH minus? OH is basic. So a lot of H plus. So I have lots and lots and lots and lots and lots of H plus. Yeah. Yeah. So if I have extra protons, where would that extra proton go? The nitrogen's full. The alpha carbon can't take anything. But that carboxylate carbon could turn into a carboxylic acid, right? All I'd have to do is add that hydrogen. So at a very acidic pH, this amino acid would be positively charged. So if you become more acidic, so if you become more acidic, you add protons. So then what if we did the opposite? So what if we did the opposite? So we're going to start back with the same thing that we had before. That's at pH 7. So now instead of pH 7, let's go basic to pH 10. So now we have lots of OH minus and not so much H plus, right? So we're not going to be adding protons. If we become more basic, you remove protons. So where could we remove a proton from? Well, what was a protonated amine could become just a plain old amine, right? So I would lose basically that. And that would be my molecule in a basic solution. So my basic solution, my net charge would be -1. So net would be -1, whereas in the acidic, the net was +1. But at pH 7, but at pH 7, it was neutral because we had a plus and a minus, which give us neutral net charge. So it's just important to know that depending on the pH, you can change the protonation state of a protein. It controls a lot. It controls a lot. That's it. That's it. That's it. If you become acidic, think there are more protons around, so we're going to add them. If you go basic, there are less protons around, so they're going to get ripped off. It's not that. Yeah. No, there are pHs at which they will pop on and off, but y'all don't have to know that. Y'all don't have to. Yeah. Okay. And we call the point at which there's no net charge, which is usually about pH 7, the isoelectric point. So iso meaning the same. So that's no net charge. Okay. So we talked about that the alpha carbon of the amino acid is chiral. And so if we look at each of the amino acids, look at your little sheet, right? If you look at each of the amino acids, every single amino acid has something different in purple, right? Except for one. And the one is glycine. So if you look at glycine, the R group is exactly the same thing as the hydrogen that comes off of the alpha carbon in the backbone. So do I have a carbon that has 4 different things attached to it? No. So that makes glycine achiral. Every other amino acid on this whole sheet is chiral. So if they're chiral, we know that we can get a D or an L configuration. We know that we metabolize sugars that were D, but we metabolize proteins that are L. It's kind of weird, but it's true. So remember how on a carbohydrate, we looked at whether the hydroxyl on the last chiral carbon was on the left or the right? Now, instead of looking at the hydroxyl, we're looking at the amine group. Is it on the left or is it on the right? And we primarily can only metabolize L amino acids, and we can only metabolize D sugars. Isn't that kind of cool? So, and I don't think y'all are ever going to have to draw it in order to determine if it's D or L. I really don't think y'all have to do that. No one has ever told me that, but there's a way that you can figure it out. So I'm not going to make y'all do anything that you wouldn't have to. All right. So anything that's different from amino acid to amino acid can always be attributed to the R group. So what we're going to see when we look at the structure of the R group, that some R groups make the amino acid hydrophobic, some R groups make their amino acids hydrophilic, right? So sometimes they're water hating and sometimes they're water loving. So if you're polar, are you going to be hydrophobic or hydrophilic? Hydrophilic. So you could be polar neutral, where you have a high affinity for water, but you're not charged, right? You're not ionic. Another word for ionic is charged. You could be negatively charged. If you're negatively charged, oh yeah, you're going to be very hydrophilic. Hydrophilic. Because that will give you an ion dipole interaction, right? If you're hydrophilic and polar neutral, you either have like hydrogen bonding going on, or you have dipole-dipole interactions, those kind of things. I guess that should be a capital H. You could be positively charged. So if you're positively charged, it's the same thing like negative. You're very hydrophilic. Again, you can make that ion dipole interaction with water. So if we take a negatively charged amino acid, you're going to look at those negative ones and you're always going to see that you have a carboxyl, like a carboxylic acid group on it that can give up hydrogen, right? So that's why it makes it acidic, whereas the basic ones are different. But it's easier to look at the, we're going to look at it. We're just going to look at it. So why do I have a blank? All right. So if you look at the amino acids, they have the name, they have a 3-letter abbreviation, they have a 1-letter abbreviation. Do you have to memorize any of the abbreviations? Nope. And the amino acids on your table are alphabetical. So you don't have to be like looking for them. If you know you want histidine, you know you're looking for H, right? So it's easy to find. So they're not grouped on this paper by functional group. Like they're not grouped by these are hydrophobic, these are hydrophilic. They're grouped just alphabetically so that you can find them, and it gives you both the 3-letter and the 1-letter abbreviation behind it. So that's nice. Okay. So when you look at these, you'll see they're just alphabetical, just like it is on the list on your extra page. But we're going to go through each of them, and you'll be able to come back and say, okay, these are hydrophobic, these are hydrophilic, and that kind of stuff. So we'll come back to that. So if you look first at the hydrophobic amino acids, all the hydrophobic, that means water fearing, right? Typically, if you think about it, you know, we're mostly water. The inside of our cell is mostly water. The outside of our cell is mostly water. So all proteins are basically sitting in a pool of water. So if you have hydrophobic amino acids, do they want to be facing out on a protein? No, they don't want to be on the surface of a protein. That would be terrible. So they're usually found buried in the interior of proteins. I-O-R, interior. So they stay isolated. They stay away from the water because they're on the inside of the protein, right? So we'll go from least complex to more complex. So the simplest amino acid is glycine. And what's highlighted in green, this is the R group. That's the green. So what it is here versus what's on your what's on your screen here. So these are both glycine. You can see they're both glycine. What's the difference between what's here in this book and what I gave you? It's just the R group's going up instead of the R group going down. But it's the same thing. It's the same thing. I just really liked how they had it. And you had all the 3-letter, 1-letter abbreviations. So that's just one thing you have to know. Okay. So glycine is the smallest one, simplest one. Then you go to alanine. Alanine, we replace that hydrogen with just a simple methyl group. That's it. 1 carbon methyl group. So I think the beginning of the alphabet, the simplest one. The next one, it's still carbons and hydrogens. So if you know that these are hydrophobic, what kind of elements should we see? Bunch of carbons, bunch of hydrogens. That's it. Because all of that is nonpolar. So all nonpolar ones are primarily, and I'm not going to say all, but they're primarily hydrocarbons. And so we see valine, we have 3 carbons, and what shape do they make? A V. That's valine, right? Then you go a little more complex and you have leucine. So valine makes the V. Leucine is a long, I call it the L for leucine, a long V. So you have that same V, but you have another carbon before you get to the V. So that's why I call it the long V. Just so that, there are some that you don't have to think about so much. Isoleucine has the same number of carbons as leucine. All we did was take one of these carbons here and stick it right there. And now we have isoleucine. So it's just an isomerization. We just kind of rearrange it. And then we get leucine. Phenylalanine is one that has a benzene ring. But remember, what's a phenol? Do you remember a phenol? It's that same benzene ring, but an OH. So this is not phenol. This is phenylalanine. So this is the benzene ring that comes from the phenol. Then we remove the hydroxyl and we put a carbon there, right? So we call it phenylalanine. That's why we call it that. Like sometimes if you know why they named it some particular way, it just makes so much more sense. Proline is one of the really cool ones. Proline is the only secondary amine. And the reason is the R group comes off of that alpha carbon, and then it comes around and it bonds to the backbone again. It's the only amino acid that does that. And it really restricts the conformation that this amino acid can take. And so it has a really big effect on protein structure. So proline is pretty nice like that. So if you look at proline, what letter does proline make? Makes a P. You have to have all these tricks because I had to memorize all of these structures. So like I had to have some tricks to help me memorize this stuff. Tryptophan. Tryptophan is very similar to phenylalanine. It has that same benzene ring, but then it also has a nitrogen group right here. So it's not completely nonpolar. It does have an amine there, but the benzene ring is so big that it kind of just like overpowers the amine. So we typically classify it as being nonpolar. Yeah. Methionine is the amino acid that is usually coded for at the very beginning of every single protein. So it's at the start of proteins. Y'all have done transcription translation. So you have like the start codon, And the start codon always codes for methionine. Yeah, like you don't have to know the codons. Like you do not have to memorize that. No. But just knowing that it's usually on the N terminus, that's a good thing. And methionine is kind of cool because it has this sulfur, but this is a very unreactive sulfur. So it doesn't do too much. Yes. It's very, very unreactive. Okay, so now we're switching gears. Everything on this page was basically non-polar. Basically. So we're going to say generally non-polar. We don't typically classify them as non-polar because you have something with a nitrogen in it, right? You have something with a sulfur in it. It's not truly non-polar, but it is, they're all hydrophobic, but generally they're not. So now we're going to switch into the ones that are polar. So polar is a big, big group, and you can have polar neutral, you can have polar and be negative, or you can have polar and be positive. So we're going to look at first the ones just that are polar but neutral. So if they're polar, they're hydrophilic, right? Which means water loving, and they're going to be basically on the outside of a protein. On outside of protein surface. So when we look at these, I want to look at the first three kind of together. So that first one is serine. Serine just has one carbon, and then it has a hydroxyl. Threonine has two carbons and the hydroxyl. And then tyrosine has one carbon, a benzene ring, and a hydroxyl. So what's common across all of them? That's right. So all three of these all have a hydroxyl on their R group. Now this is really, really important because they can all be what's called phosphorylated, can all be phosphorylated. So do you remember in our different functional groups paper, we said a phospho group, right? A phosphate group. Like you had an R and you had a phosphorus and an oxygen and an oxygen and an oxygen. So we can take this group right here, take it off of the R and put it on the oxygen of any of these amino acids. And so phosphorylation events are like molecular on-off switches. On switch. You got to think about it. You're going to have like a ton of proteins in the cell at any given time, like a ton. And if they were all working all at the same time, it would be ridiculous. It would be a mess, right? Nothing would get done. So you have to have a way to basically have the protein there, but turn it off or turn it on when you want to turn it on. And so that's what phosphorylation is. It allows you to turn on and off protein activity, which is really, really cool. I think lots of things are really, really cool. All right, cysteine. Cysteine is a really cool one because cysteine can do something that nobody else can do. Cysteine can form what are called disulfide bonds. And disulfide bonds are really, really important to keep proteins in their three-dimensional shape. Because you got to think about, this is the way I think about a protein. I think about a protein as a shoestring, right? And when we make it, it's this long shoestring. But that's not how the proteins looked in that video we watched, right? They were all like folded up and in these like weird conformations and all kind of craziness, right? So if you got to think, okay, this protein has to maintain this shape. How does it maintain this shape? Well, one of the ways that you could do it is you could hold it together. What's called a disulfide bond. So if I had two cysteines, so I'm going to put another CO minus, an NH3, right? Okay. And then I'm going to put my green. Oop, I forgot my hydrogen. Don't forget your hydrogen. I'm going to put a CH2, and then I'm going to put an SH. So what will happen is each of these sulfurs will lose the hydrogens. They'll lose those hydrogens, and they'll form a bond between the two sulfurs. That is the disulfide bond. And it creates like a bridge that locks the protein into a particular conformation. It's really strong because it's covalent. Methionine. It's that the sulfur is bonded to two carbons in methionine. So in methionine, it's bonded to two carbons, which makes that sulfur super unreactive. But if we attach, instead of two carbons, one hydrogen, that hydrogen could be ripped off, and then it could form a bond with something. So carbons are basically, they're like locked in place. They're not going anywhere. But hydrogens can pop on and off of molecules all the time, right? Yep. So that's why. That's a great question. So this sulfur is very reactive, where the methionine sulfur is not. That's great. That's great. Okay, so then we have these last two, asparagine and glutamine. So asparagine has kind of two functional groups kind of going on, right? We have just a regular old carbon, and then we have a carbonyl, and we have an amino group. So do you think those would want to interact with water very much? Sure, like this could have hydrogen bonding, right? Then the oxygen could have dipole-dipole interactions with water. So very, very hydrophilic. So what's the difference between asparagine and glutamine? I have the same carbonyl, I have the same amino group. The only thing that's different is here I have one carbon, here I have two. So the length of the chain is a little bit longer in glutamine, but very similar in asparagine. So why do I end it in INE? Because they're amines. So the amines are referring to these amines and not those amines. Because we have asparagine, tyrosine, threonine. Correct. Correct. So just want you to be careful about that. The backbone, NCC, the backbone NCC. Yep. And that backbone is this right here. All of those atoms there. Yeah. Okay, so those are the polar neutral amino acids, right? They want to hang out with water. They're happy to hang out with water. Then you have the charged amino acids. Are they still water loving? If they're charged amino acids, I could have ion dipole interactions with water, right? Interactions with water. So that means that my charged amino acids are also hydrophilic, right? So when we talk about the ones that are negatively charged, the negatively charged ones, those are our
acidic, ACIDI, acidic amino acids, and then the positively charged ones are the basic, BASIC. And we're going to look at some similarities in their R groups. So if you look at the acidic ones, what functional group is that? Carboxylic, BOXYL. It's really a carboxylate that came from a carboxylic acid. So this is why we call them acidic, because when we put them in water, they donated that proton. And so they're acidic, right? So the only difference between our two, which is aspartate and glutamate, which is very similar to asparagine and glutamine, right? What's the only difference between asparagine and glutamine and aspartate and glutamate? Well, right, exactly. One's a carboxylate, is the amine. Well, amide, yeah, it's an amide on the end. It's an amide on the end. Yep. So if you look at the carbons are the same. Oh, we're going. All right, we're done. We're done. We were just getting carried away.